Hey all, so today we're going to be looking at D2.3, which is HL content related to meiosis. We're looking at page 710 to 18. Continuity and change is continuing to be our theme, but for this one, we're describing in detail how crossing over and independent assortment impact gamete variation. So firstly, in interphase, DNA is replicated, and that means it makes a copy of each parent chromosome. And we've seen this in the other video, but these are homologous chromosomes. So they're both chromosome one from mom, one from dad. And when they duplicate, they're going to make a copy of each other. But this one, the blue one here, is an exact copy of the blue chromosome that came from, let's say, your dad. And the pink one is an exact copy of the pink chromosome that came from your mom. But the two of them, the reds are sister chromatids and the blues are sister chromatids, but the two of them to each other are homologous chromosomes because they're both chromosome 1. And so in prophase 1, what happens is synapsis, and they line up to make something called a tetrad or a bivalent, and then they're held together by proteins called cohesion loops. And then the non-sister chromatids, so meaning the blue one and the pink one, because the blues are identical and the pinks are identical. So the non-sister chromatids, a blue and a pink, can cross over so that they can exchange alleles, potentially different alleles. And that break point where they've made the swap is called a chiasmata. And that can happen at different points along a chromosome multiple times sometimes. So crossing over can decouple linked genes. What that means is, firstly, we need to know what a linked gene is. They're close together on the same chromosome, and they often cross over together. So if they're on chromosome 1, like this one, these are linked, they're close to each other. An example of a linked gene would be red hair and freckles. Typically, people with red hair have freckles, and that's because they're crossed over genes. They're linked genes cross over together. But then we could have non-linked genes. That would be if it's on chromosome 1 and a different gene on chromosome 2, those would not be linked. Or if they're really far apart on the same chromosome. And so crossing over can decouple linked genes. That means there could potentially be a break here between gene 1 and gene 2. It's potentially unlikely, but it could happen. And so that would decouple, meaning prevent them from being stuck together, linked genes. So with crossing over, that creates variation in gametes, but also so too does independent assortment. And so the direction that the chromosomes line up in metaphase and split is random. There's a 50-50 chance of the chromosomes lining up with the pink facing one direction and the blues facing the other direction. It just is totally random. And then therefore they will split totally randomly. And the orientation of one chromosome is not going to affect how the other lines up. So independent assortment and segregation of alleles. So segregation of alleles means that, so this one here, this is in metaphase 2, you can see that there's crossing over that's happened here, but that crossing over piece is not going to affect the ability of the chromosomes to transport. And then additionally, segregation of alleles, meaning any particular allele could cross over. However, if genes are linked and they're close together on the same chromosome, this will affect whether or not the alleles will cross over randomly. And so linked genes will typically cross over together, which means that the segregation of alleles is not random because linked genes are crossing over together. So we've already looked at monohybrid Punnett grids, but dihybrid Punnett grid is something that we need to know about for HL. And this is a Punnett grid with 2 traits by 2 different genes. And so in this image on the right, we are looking at seed color and smooth versus wrinkled surface. And so we have heterozygous P parents, and you can see that they have an S gene and they have a Y gene, and the S gene is heterozygous and the Y gene is heterozygous. And so the possible gametes that these 2 P parents could make are going to be the same, but there's 4 possible gametes. And so we need to include both genes, the S gene and the Y gene. And so we could have capital S, capital Y. We could have capital S, small y. We could have small s, capital Y. Or we could have small s, small y. And this is just how the gametes would potentially be generated. And there could be options for any of these possible gamete combinations. And so if we are going to create the Punnett grid, then all we have to do is write into the squares the 2 different characteristics, the gene for S, and so we keep the S's together, and the gene for Y, we keep the Y's together. And so there would be 4 letters in there. And so you can see here, there are 9 smooth yellow pea plants, even though they're sometimes heterozygous, sometimes homozygous dominant. And then there's 3 smooth green peas, and then there's 3 wrinkled yellow peas, and then there's 1 wrinkled green pea. And so the Mendelian ratio for this is 9 to 3 to 3 to 1. And so if everything is crossing over randomly, this is the ratio that we should see. But if things are not crossing over randomly due to linked genes, then we're not going to see this ratio. So you can have linked genes on typically for DP purposes, we're going to see this only for autosomes, so body cells, not sex cells, but they could be linked for sex cells. But sex linkage is different, and it relates to specific genes on sex chromosomes. So we're not going to refer to that throughout DP. But if they are linked, then what's going on is that they're not going to cross over randomly, and you're not going to see the Mendelian 9 to 3 to 3 to 1 ratio showing up. And so Mendelian ratios for single trait are 3 to 1, and for dihybrid Punnett grids, it would be 9 to 3 to 3 to 1. And Thomas Hunt Morgan of the fruit fly sex-linked genes traits discovery, he also discovered that gene linkage occurs. And what Mendelian ratios did, well, so he was expecting 9 to 3 to 3 to 1 for Mendelian ratios in his fruit fly babies, but they didn't show up like that in the F2 generation. So when we had heterozygous crosses, or when he had heterozygous crosses happening between F1 generations to produce the F2 generation, the Mendelian ratios didn't show up. And this was due to gene linkage, which results in a higher than expected number of parent phenotypes and a small recombination portion of recombinants. And that means that crossing over isn't happening that much, and those characteristics are sticking together. If the offspring outcomes are not Mendelian, it means that the linked genes are acting on the gametes. And so here we have a diagram, an image from your textbook on page 717. And the lines here, this is how we would draw linked genes. And so the lines represent the homologous chromosomes and the traits for the 2 alleles. And so in this case, we have the parent genotypes. And so parent, whatever this parent, let's call it mom parent, can produce big C, small n gametes, and parent number 2 can produce small c, big N gametes, because that's all there is there. And so you end up with the F1 generation, big C, small n, and small c, big N. Okay, so what's going to end up happening here, if crossing over is happening, then we're going to have big N and small n switching places. And if that happens, then we could have recombinant possible gametes. So the normal gametes, parent gametes, would be big C, small n. So we see that here, big C, small n. Or we would have small c, big N, small c, big N. Those are the parent gametes. And then if recombination is happening, so crossing over is happening, the N's will swap locations here. So we can't swap C and N because those are not the same gene. We could swap C and C. It doesn't matter. Just pick one and swap it. So then a recombinant gamete would be big C, big N. So that would be a recombinant gamete, and then small c, small n. And so that's a recombinant gamete. And so here we're doing a test cross where we have small c, small n recessive traits, And then they're going to crossbreed and see what kind of seeds were produced. And so we can see here that we have 21,000 pea babies being produced from the parent gametes. And then we have this tiny little amount being produced by the recombinant gametes. And so what this is telling us is that the color and the shape of those seeds is linked. And so it's very rare that we're going to end up with recombinant gametes being produced. And this test cross was being done for the F2 generation. So crossing over, homologous chromosomes attached to make a bivalent, and then non-sister chromatids exchange genes at the chiasmata. Independent orientation means the chromosomes can line up any which way in metaphase 1 and metaphase 2, and they have a 50-50 chance of splitting to the opposite pole. And then we would also have that in non-linked gene situations, crossing over can also happen completely randomly, and that would be called segregation of alleles. The Mendelian ratio for a monohybrid trait would be 3:1. And dihybrid traits would be 9:3 to 3:1. And if you're not seeing a Mendelian ratio, that indicates that the genes are linked, meaning that they're less likely to cross over to make recombinants. And if we looked at this diagram here, we can see these are linked genes because they've just drawn them on this line like this. And so we have small c and waxiness, and big C and waxiness. And the small WX is obviously the recessive trait. So this is the mum gamete, and these are the dad gametes. And so dad you can see that C's and WX's are the same. That means dad can only make one type of gamete. So he's only making small c, small WX as gametes. But mum could make small c, big WX, big C, small WX, and then we can also swap it. And so in the outcome of this mating, we would have small CWX, that's from mum, and then this is dad's gamete. Dad's gamete is always on the right here. And we would also potentially have big C, small WX here. And so these are the non-crossovers. So no crossing over has happened. These are called the parent gametes. But then in mom's genes, crossing over has happened potentially. And so small c, small WX, and big C, big WX are potential recombinants that would be possible in those seeds. So this is just another example. Some students do have challenges with this. So if you're struggling with this, please definitely ask for help with this. Okay, that's it for this video. Thank you very much for watching.